literally told me a minute ago, so I'm coming.
Well,
Is it
all, everybody, everyone. Hello, Dr. Biology here, and this is Cell Biology Revision. Thank you. Sorry, I was just doing. I'm going to record it. Okay, good. Right, and well done for being here. Right, should we get started then? Good. Okay, I don't intend this to be longer than 30 minutes, probably 25 minutes. So cells. So we're first of all going to talk about types of cell, okay? And we're going to first talk about, up here, we're going to talk about eukaryotic cells. So eukaryotic. Okay? And there are two groups of cells you need to know a lot about. And what are they? Two types of eukaryotic cell, what are they? Yeah. Yeah, nice and simple. So we'll go for animals. So animal. Now, so animal cell, here we go. So I'm just going to draw a very simple animal cell with the particular parts in it that you might find. In fact, I think I'm going to make that bit bigger. Okay, and then in it, I've got little things in it as well. Okay, so let's talk about the parts then. So what's that, do we think? Fraser? It is the nucleus. Well done. So nucleus, that's basically where the DNA is held, and also that controls the cell. Okay, this part here, please, the outside. No? Okay, yeah. Cell membrane, well done, very good. So that's the cell membrane. That controls what goes in and out of the cell, so in and out. This blue thing, I'll give you a clue, it's where respiration occurs, yeah. No, where respiration occurs, yeah. Mitochondria, very good. Mitochondria, so that's the release of energy. Good, mitochondria. And then the little red dots, who said, yeah, it is the ribosomes. Do you know what happens in the ribosomes? Yeah, it makes proteins or synthesizes proteins. Very good. Okay, and there's one last thing, is the cytoplasm. So that is where chemical reactions occur. If you're doing foundations here, they love you to label bits, label cells like that. Right, okay. So plant cell, I'm going to do a plant cell here. Now, plant cell is pretty much the same, okay, except it's got a few differences. We'll talk about what those differences are in a minute. So, should not sing. Yeah. Right, so it has all the same parts as the animal cell, but it has three main differences. Can anyone tell me one of them at least? Fraser? Well done. So we're going to go for the permanent vacuole first of all. Permanent vacuole. Well done. Okay, that's one difference. Another one, yeah. chloroplasts, well done. So chloroplasts are where photosynthesis occurs. So chloroplasts, and there's one last thing. Yeah, cell wall, well done. Very good. Now, they do like you to know the differences between animal and plant cells, and that's the difference, basically. There's more features in a plant cell than an animal cell. Good. Okay, so we're going to look at the second type of cell now, over here, and the second type is prokaryotic cells. Prokaryotic. Should be an A. Well, I'm going to draw a prokaryotic cell, not very well. Okay. It's going to have bits of DNA in it. Sorry, it looks like a sperm. Actually, it shouldn't look like a sperm. I'll do. There you go. Ooh. Right, okay, so this is a bacteria, believe it or not. Okay, it is a prokaryotic. So bacteria, the big difference with bacteria are that they have loops of DNA. So these are called plasmids, which is a loop of DNA. So it doesn't have a true nucleus. So there's no nucleus. That's the big difference. But it has things like a cell wall, different to a plant cell. And it does have cytoplasm. It does have ribosomes as well. Okay, but that's the big difference. Now, in terms of size, okay, these are about 10 to minus 1 smaller than plant and animal. Okay, 10 to the minus 1. So that's kind of structures. We need to talk a bit about specialized cells. I'm not going to do very much with specialized cells, apart from tell you which ones you need to know. There are 6 specialized cells you need to know about, okay? And you would have done some research on them. All right, and I'm going to tell you what they are. So here we go. So I'll just do it down here. So first one is sperm cell. What's the function of a sperm cell? Well, it fertilizes the egg. Fertilizes the egg. Okay, it's got a tail, so it can swim, etc. It's got enzymes in the head as well. Red blood cell. What does a red blood cell do? Yes.
Does it transport nutrients around the body?
No. Red blood cell transports oxygen. Transports oxygen, has no nucleus because it needs to pack in the oxygen. So it's got no nucleus. Okay. You also need to know about muscle cells. Okay, so muscle cells, they've got lots of mitochondria. They contract and relax. So they contract and relax to allow movement. Okay. Nerve cell. What do nerve cells do? Yeah.
Impulse electrical signal.
Or they carry electrical impulses, yeah. So they carry electrical impulses around the body, is what they do. Good. And then we need to know about two plant cells, which is the phloem and the xylem. Okay, and you would have done that when we talked about organization in plants. So the phloem transports sugars up the plant, or glucose, well, transports sugars, let's say, up. And xylem transports water up and down. Oh no, wait a minute. I think I've got that wrong. And cut. Okay. No, sorry. Just put transport, right. So right, 3, 2, 1. And the phloem, it transports sugars, whereas the xylem transports water. Okay. They sometimes like to talk about root hair cells, but I'll talk about that in a minute. Right, I need to speed up. Okay, let's talk about microscopes next. So, there are two types of microscopes we need to know about. That is light, okay, and electron. I'm going to write a couple of features of each very quickly. So, a light microscope uses lenses, okay? So, I'm going to put a different color actually. So it uses lenses. Tends to be transportable. So it's small. You can, it's about times 2000 mag on them. Whereas electrons, they are not controlled by light, okay? They've got a much higher magnification. So times 1 million at least, probably more. They've got a much higher resolution, so that means you can actually see the pixels. So they've got a shorter wavelength of light. Well, it's not light, the electrons has a shorter wavelength. Okay, so calculations, so calcs. You will have to do calculations, and this is a good way to remember it, is the I-am triangle. So I equals image, which is in millimeters. A is the actual size, usually in micrometers, and we'll talk about that in a minute, and M equals magnification. So you measure in millimeters, okay, and you sometimes need to convert it into micrometers, okay? So that was it, yeah, millimeters. So if you have something that, right, okay, so if you have something that is 44 millimeters, okay, I can convert it into micrometers by times it by 1,000. So that equals 44,000 micrometers, which is quite a lot. Okay, so you can times it by 1,000 to get micrometers. So here's an example. Here's a cell. Okay, I'm going to measure it in millimeters. So I measure it in millimeters, and it is, I don't know, it's 36 millimeters long. Okay, but the magnification is times 500. Okay, so how would I work out what the actual size of that is? Well, I know the image, I know the magnification, I want to find out the actual size. So put my hand over there, and it's I divided by m. So I divided by m, so that equals 36 divided by 500, which is 0.072. But that's in millimeters. So I want to convert it into micrometers. So I times it by 1,000, and that gives me 72 micrometers. There we go. But you'll never have to write it. Right, okay, so we're going to talk a bit about chromosomes and genes. Okay, so we're going to talk about genes. So in the nucleus, so found in the nucleus, so you have chromosomes that hold, well, what do they hold? Well, they hold genes, which holds DNA. Oh, forgotten the nucleus. So let's stick the nucleus. There we go. So we need to know about how are new cells made? So new cells. So what happens is mitosis. Okay, so there's 3 stages I'm going to talk about. One is DNA replication. So I've got chromosomes. So the growth of the cell. I'll just pause. Got 10 minutes left. Well, no, I've got 10 minutes left. Okay, then what happens is that the chromosomes, they split themselves, okay? So you get them splitting. In fact, actually, that's the original. So chromosomes go to each side of the cell. Each side of the cell. There we go. And finally, what happens is that they then separate out. So you get what we call cytokinesis. Okay, so they go either way. And you get cytokinesis. So you're producing you're producing daughter cells, produce clones. Okay.
So,
Yeah.
what's the last bit? Why is the X?
Sorry, that.
Yeah.
So they've joined up. So in fact, actually, sorry, that's incorrect, isn't it? It's one of those and one of those. There you go. All right. So it makes, you can see that DNA is replicated. Chromosomes go to each side of the cell, and you get cytokinesis. Right, I'm going to go back up here. Okay, I've nearly finished. So Cytokinesis. If you can't do that in an exam, just say that the cell splits. So you produce a clone. CYTOKINESIS. Great, cool. Right, let's go up here. And I've got two more arrows to go. I've got stem cells and then transport. Stem cells. Okay, stem cells. So they're made in embryos. You also have adult stem cells. Okay, and they are what we call undifferentiated. Undifferentiated. Okay. Okay, why are they exciting? Because they can make any type of cell. Make any type of cell from embryos, but adult stem cells, you can only make a few types of cells. However, what they are doing now is something called therapeutic stem cell research. So therapeutic cells, where they take adult stem cells and they basically zap them with electrical impulse. And they start to behave like that, like an embryo. Just a quick thing here, plants have stem cells too. Plants, and where they are produced is the meristem. Sounds something like out of Lord of the Rings. It's meristem. Okay? Meristem. Right, okay. So there are pros and cons of stem cells. Okay, and I'm just going to go through a couple of pros and cons. Okay, so pro, con. Pro is you can relieve suffering. You can cure things that we couldn't cure before. You could argue embryos would be thrown away anyway from IVF treatment. So why not use them? What's some cons? Well, some cons are that could an embryo be life? So embryo, you destroying a life. Never use the term playing God. Okay, we are playing God. That's not a biological term. So embryo destroying a life. Okay. Also, we don't understand, so lack of understanding. It's expensive. And also viruses could be transmitted. Viruses transmitted by stem cells. There's lots of cons. I'll just give you a moment longer to do that. We'll get on to the last bit I'm going to do today. I'm going to do this as simple as I possibly can. So I'm not going to spend a lot of time on this. So you're going to have to do some extra revision. If you're watching on DrBiology, check out my playlist in the comments, okay, for more information. Right, so transport. Okay, 3 types of transport. We'll talk about diffusion over here. Okay, so diffusion. Very quickly, gas, particles, or molecules, they move from a high to a lower concentration. It's passive, so no extra energy, no energy required. There we go. Osmosis. Let's do osmosis here. So it's water. It's water that is transported. Okay. Water moves from a dilute to a more concentrated solution. There's a very good video on that on DrBiology. You don't understand that. And it has to travel through a partially permeable membrane or semi-permeable membrane. Again, no energy required, same as diffusion. So I've put it over there with them. So no energy required. And then the last one is active transport. Okay. So active transport is basically, again, it can be usually molecules, so like mineral ions, stuff like that, gases, sugar, glucose. But the big thing here is it goes from a low to a high concentration. And most importantly, energy is required from ATP, from the mitochondria. So energy is required. You can just put molecules, so minerals, glucose, stuff like that. Good. Okay, so that's pretty much everything. The only thing I have missed out, and you need to probably look up, is gas exchange. So actually, sorry, can I just do this one thing? Gas exchange. Just going to do a very quick thing. Right, 3, 2, 1. So gas exchange. 3 things you need to remember, guys. 3 things. For good gas exchange, you need a large surface area. You need thin membranes. So thin membranes, so like the lungs, you've got the alveoli, 1 cell thick, capillaries, 1 cell thick. And also you need a steep concentration gradient. So high to a low concentration. So when you breathe in, lots of oxygen in your lungs, but not so much in your blood. And that brings us to the end of our revision for today. Thank you. There you go. All right. I will put that video on DrBiology as well, so you can go through it. Okay. Quite a lot of information, but it's just a good summary of what you need to know. Well done, guys. Thank you very much. Thanks for coming.